Today's lecture. So in the past few lectures we talked about some of the techniques and some of the ideas about genome-wide analysis that analyze the level of proteins and RNA and metabolites and microbes, etc. Right? So what do we do with that kind of data, right? So there are two ways to approach a lot of scientific problems. Of course, there are more than two ways, but two ways to approach scientific problems is you go one at a time with a hypothesis, with a hypothesis in mind, and you go and test your hypothesis, right? And the second way to approach a problem is you collect the data, which allows you to build hypothesis on, right? So some of these omics approaches are essentially a way that enables us to analyze data more complete. What I mean by that is it allows us to analyze our bodies, organisms, as systems. Okay, so what do I mean by a system? Our body is a system. What I mean by a system is interaction between its components. Okay, so the components of our bodies are genes, right? That's the information, the proteins and the RNA products, functional products that it makes, and some other molecules and etc. So everything in our body is a component of our body, and a system is the interaction between the components. Okay, so let me just go back here. Okay, so here are the components in a biological system, like genes, RNA, proteins, small molecules, metabolites, etc. And the interactions are what makes these components function, right? So, bless you. The protein-gene interactions, like transcription factor to gene interactions, controls gene expression. Protein to RNA interaction are essential for post-transcription regulation. Protein-protein interactions are the basis of all the protein complexes and molecular machineries that do all the jobs, right? Like the transcription factor machinery, transcription machinery, or replication machinery. And protein metabolite interactions allows our bodies to function and essentially giving us energy and everything else, right? So the omics approaches lets us measure all of these components, right? And we have molecular biology tools that allows us to measure these interactions. For example, some of the techniques that we have learned is the ChIP-seq technique that allows us to determine which proteins, which transcription factors bind and regulate which genes, right? Similarly, there are techniques such as RNA immunoprecipitation followed by sequencing that allows you to determine which RNAs are bound and regulated by which proteins. And today we're going to talk about another technique called yeast-2 hybrid that allows you to measure interactions between proteins, okay? So why do we bother with network analysis? And what can it tell us about biological systems? So to answer these questions, let's first define what a network is, okay? So a network is made up of nodes and edges that connect them. So each node is in a network is like this, and there is an edge. There's a relationships that connect these nodes, okay? So you, of course, know what a social network is, right? So in a social network, you are the nodes, and your social interactions are your edges, right? So your friends, your family, your neighbors, etc. are your social interactions. In a highway network, right, the cities would be the nodes, and then the edges are the highways that connect between these nodes, right? And in a gene network, in a gene regulation network, the transcription factors would be the nodes, and then their target genes also would be the nodes. So the relationship would be that transcription factors binding and regulating the transcription of the target gene, that would be the relationship. So that would be the edge. Okay? That makes sense? All right. So how do we actually visualize this kind of network data? We visualize this data like this. So what we do is we put the nodes in little circles like this, or whatever you want to do, just a little a shape to show you what it is. And then the edges we show by these lines connecting the nodes, okay? And the degree of each node is related to number of edges a node has. So if in a social network, if you have a friend who has thousands of friends, that would be that your friend has a high degree of interactions because he or she has lots of friends, right? And then if there's a loner that has very few interactions, that would have a lower degree of interaction, right? And so the nodes that have a lot of interactions, we call them hubs because they connect a lot of different nodes to themselves, right? So that's called a highly connected node. All right. So in various examples, what we see is in general two types of networks. One is called a random network, and the other one is a scale-free network. So the difference between these is this. In a random network, it looks like the highway system in the US, okay? So each node has few connections, and these connections are basically appears random. It's not random in the highway, but what this means is that in this network, you have these nodes that are making connections to their nearest nodes, and they have few connections. And a scale-free network would be for example, the airline network. So in this kind of network, what you have is that each node makes connections, and then there are some nodes which make many, many connections, right? And so these are the airline hubs, so it would be Chicago, New York, Atlanta, or etc. Does that make sense? So in this random network, you have every node having few connections, and in a scale-free network, you have a small number of nodes that has lots of connections, lots of edges. All right, so how do we distinguish between a random network and a scale-free network in math terms? We distinguish them by plotting the distribution of the number of links in a histogram. So if you plot the number of nodes that contain a given number of links that are in the x-axis, what you will have is in a random network, you're going to have an average number of edges per node. So you're going to have a random distribution of these edges, okay? So a typical node is going to have a certain number of links, a certain number of edges. In a scale-free network, what you're going to have is you're going to have a power law distribution of node linkages. What that means is that for what you're plotting is you're plotting the number of nodes that have a given number of links, a given number of edges, okay? So in this kind of flight network, you're going to have a lot of nodes with very few edges. Okay, so you're going to have lots of small towns across the U.S. that has a single airport, and the flights that are coming out of this airport is going to be going to a single hub, right? So a small town near New York, like Allentown, is going to have one flight to New York and one flight to Philadelphia. So very few nodes, right? And then you're going to have a small number of nodes, which will be the hubs. They're going to have lots of links, right? So like New York, you have flights to all over the place, right? So when you plot that, the number of nodes and the number of links, you're going to get this power law distribution. and if you take the logarithm of the y-axis scale, then you can, and the x-axis scale, then you can change this distribution into a linear distribution. Make sense? All right. Okay, so the scale-free networks are much more relevant to biological systems than random networks. And here are some other examples of scale-free networks. So the cellular metabolisms is a network that's scale-free. So the nodes are the molecules involved in burning food or energy, and the links would be the participation of these cellular metabolism in biochemical reactions. In Hollywood, there are a small number of actors that work in many, many movies, right? So, and then there are a large number of actors that work in very few movies, and other examples here. All right. So why am I talking about these two types of networks? The way that these two networks function are very different, and one example for that is that their robustness is very different. So what that means is that the random networks, they can be fractured. Their function can be altered by failure to any node, right? So let's say that there is a very small town between New York and Chicago on a major highway. And then in that small town, there was a fire, and that closed the highway, right? And what that's going to do is it's going to affect all of the transportation between New York and Chicago. So the network is going to be kind of disrupted. And all the trucks that are going from New York to Chicago are affected, right? And they're going to other cities. So all of the network is going to fall apart because there's going to be no way to bring goods from New York to the west of the country, right? In a scale-free network, this kind of networks are robust to most node failures. What that means is that, for example, a small town between New York to Chicago, if their airport actually shuts down, we're never going to feel it, right? Because most of our flights from New York is going to just fly right over that little airport, because that little airport has few edges, few links. Its lack of function is not going to affect the whole network, right? Unless in a scale-free network, the damage is done to the hub, right? So whenever there is a snowstorm in Chicago or Detroit, which are both hubs for airlines, you're going to have all of the planes delayed across the U.S., right? Because they're connected to many, many other airports. The absence of them affects the whole network, right? So in a random network, attacks to single nodes are going to fracture the whole network because every node basically depends on each other, right? And in a scale-free network, attacks to both of the nodes is not going to matter for the network, except if the attack is to a hub, then it's going to affect the network function. Okay? And so this was actually known to biologists for decades. That is, the biological systems are scale-free networks because a lot of the mutations, so most of random mutations in a biological system, in an organism, does not affect any function. Okay? So, but mutations affecting any of the hubs are lethal. So we have 20,000 something genes. Most of the genes, if you knock them out, are not lethal. But there are a handful of genes, if you knock them out, they're lethal. Okay? So basically, this kind of scale-free networks and analysis of data and thinking of this in this kind of thinking frame provides a quantitative explanation for a phenomenon well known to biologists. Okay? All right. So this is how we think about networks. Networks are interactions between components, and to build a network, we need to actually measure these interactions, right? So one of the biggest interaction that is functional in cells are protein-protein interactions, right? So proteins binding each other. We talked about this over and over again. Proteins forming complexes. For example, the formation of the pre-initiation complex at the promoters. Interaction between sigma factor and RNA polymerase. These are very important interactions that have important functions, right? So then we need to have a method to measure these protein-protein interactions. And how do we do that? So we want to understand the network of physical interactions between all proteins in the cell that carry out the cellular functions encoded in the genome. And the technique that we can use is yeast-2 hybrid. So in this technique, we're again taking advantage of the transcription factors having two separable domains, okay? So what we're doing is, here I'm going to give you one example. Let's say you are interested in one protein and you want to know what all the other proteins in the cell does this protein interact with, okay? So what you do is you prepare a bait vector. So your bait is your protein, and you're going to fish out all the interactions that it makes, okay? So what you do is you clone your protein, the DNA sequence that's encoding for your protein, which is the bait gene, and in a way that fuses this protein to the DNA binding domain of a yeast transcription factor, okay? So when this vector is transformed and expressed in yeast cells, it's going to produce a protein where the DNA binding domain of the yeast transcription factor is going to be fused to your bait. Okay. And this vector also has a trip marker, which will allow you to select for cells that contains this plasmid, okay? In the same cells, you transfect a library of proteins that are also cloned in such that you have every protein from your organism that is fused to the activation domain of a yeast transcription factor. So once this vector is the cloned sequences are expressed, in the same cell you're going to be producing a protein, right? All the different kinds of proteins that are fused to the activation domain of the yeast transcription factor. So what you're testing is you're testing the interaction of your bait with this fish, okay? So interaction of your bait with all the possible proteins that are in your library. Any questions about this? All right. So what's the readout? The readout is this. So in your cells, you also have a reporter construct, okay? And your reporter contains the upstream activating sequences that your DNA binding domain will bind to, okay? And then that's going to, if it binds and activates the expression of this His gene, these yeast cells are going to be able to grow in plates that don't contain any histidine, all right? So essentially, if this happens, that is your DNA binding domain binds to your upstream activating sequence, and DNA binding domain is fused to your bait, right? And if this bait is interacting with your protein of interest, right? Proteins in the library, all the different proteins in the library, which is also fused to the activation domain of the yeast transcription factor. This interaction is going to bring the activation domain to the vicinity of the promoter, and that's going to increase the transcription of the His mRNA. So these yeast cells are going to be able to grow in media not containing any histidine. Any questions about this? Yeah.
So how would that determine what specific proteins the bait construct interacts with?
So you actually know what protein is in your library. So when you make your constructs, what you're going to have is you're going to have your bait protein construct, right? And then in your library, you make this library. And you know in your plasmid 1, you have protein 1. In your plasmid 2, in your protein 2. In your plasmid 3, you have protein 3, etc. And you transform the cells in together, right? You're going to have cells that contain these two, or these two, and or these two. Yeah?
Okay, so the individual plasmids don't each contain the entire cDNA.
Individual plasmids contain the entire, not all of the possible proteins, right? It's going to have the cDNA for one protein each. Does that make sense, guys? Yeah?
Would you have to do like successive trials in order to know which proteins?
Exactly. It's a pairwise interaction, right? So if your library has 1,000 proteins and you have one bait, you're going to do 1,000 different tests, right? Because you're going to do that in pairwise transformation. Any other questions? Okay. Very good. All right, so here's a little example of this. Okay, so let's see that we're trying to look at pairwise interactions between 4 proteins. These proteins named A, B, C, D. And instead of looking at one proteins, like protein A's interaction with A, B, C, T, we're doing in a pairwise, right? So we're looking at the interaction of A with A with B, A with C, A with D, and then B with A, B with B, B with C, etc. Right? So it's an interaction matrix. So we're trying to do this using yeast-2 hybrid. So when you transform your bait vector with your fish vector one at a time, right? To make sure that your cells contain both of the vectors, you grow these cells in plates that are missing the trip and the leucine amino acids, okay? So these cells can only grow if they contain the bait plasmid that contains the trip gene and the fish plasmid that contains the leucine gene. So when you plate your cells that contains each of these plasmids in pairwise, your colonies are going to grow, and you're going to see colonies on your plate, right? All right. So your reporter is the expression of the hist gene. If it's expressed, the yeast colonies are going to be able to grow in plates not containing histidine. And so when you take these colonies and you replica plate them, what that means is you take yeast cells from each of these spots and then you plate them onto another plate. Again, you want to make sure that both plasmids are there, so minus strip and minus Lu. And you're testing if your reporter is expressed, so these plates also don't contain any histidine. So whenever you see a colony growing, then you know that in this colony, the presence of these two plasmids actually activated the transcription of the hist gene, right? So what that means is your bait gene actually interacted, your bait protein actually interacted with your protein that is in your fish vector, right? So when you see a colony then in the minus strip, minus Lu, minus hist plate, you know that the protein A interacts with protein B. It also interacts with protein C and D, right? And protein B interacts with protein A. It interacts with itself, but it doesn't interact with C and D. C interacts with A and D interacts with A and C. Does that make sense? So that's how you read these interactions. Now let's convert this data into a network, okay? So in this network, what we have is that we have each of the protein as a node, and the physical interactions as measured by yeast-2 hybrid is the edges, okay? So what you do is you put all your nodes into the network, and then you put an edge whenever you see an interaction. So I see an interaction between A and B, so I put an edge between them, connecting them. And the A is also connected to C and D. B is connected to itself, right? So you're going to put a little edge to itself. What that means is maybe it homodimerizes, right? So proteins interact with themselves and dimerize. And then C and D can interact with each other, etc. So you create these protein interaction networks using the data, right? So what's the nodes here? In this network, protein interaction network, what are the nodes? Each protein. And what's the edge? Interaction. What protein appears to be a hub in this network? A, right? So that's essentially the gist of this protein interaction networks. Okay, so this was a kind of a simple example, right? In fact, when we do yeast to hybrids looking at the whole interactome, we call this an interactome because it contains all the interactions, right? So the actual plates look something like this, all right? So this is the data from a yeast interactome in which only a single protein's interaction was analyzed. So in this case, the bait was PCF11, which is a component of the pre-mRNA cleavage and polyadenylation factor A. And the prey is the 6,000 different yeast proteins, okay? So from this interaction, so this is plates containing the histidine, and these are plates not containing histidine. Only 11 different colonies grew, right? So you know that there are 11 interacting proteins, and then 3 of which were already known to form a protein complex with PCF11. So this is one protein against each other, against everybody else. You can repeat this analysis in that you, as a bait, you use every single protein within that 6,000 yeast proteins. Then you would be measuring 6,000 times 6,000 interactions, right? So these are pairwise interactions. So the researchers tested 36 million interactions, and they found 957 interactions between about 1,000 proteins, okay? So then they took this and converted into a network map. So this was the article from 2000, a comprehensive analysis of protein-protein interactions in yeast, Saccharomyces cerevisiae. How does the interaction network look? So now we're looking at a much more complex network, right? So these are all the genes that are shown here in green and red and all of these circles. And then what the researchers did is let's color all of these genes based on our information that if you have a knockout of this gene, whether it causes lethality, slow growth, no phenotype, or it hasn't been tested. Okay? So you have all the genes here, and then they're color-coded for whether they're lethal or not. And then what you can see is that you don't see much, right? It's kind of a hairball. Actually, for a very long time, a lot of labs that did this network analysis would call these hairballs. But some of the things that you can see is, for example, here's a gene that is a hub, right? It's connecting to many other genes. And here's another gene here, again, appears to be a hub connecting to many other genes. Okay, so let's now test whether our initial explanation, which is that few genes, the genes that are more connected, the genes that have more edges, more links, are they more likely to be lethal when deleted, right? Because that's kind of what we said. So this is the analysis what they did, okay? So we call the genes that are, when deleted, cause lethality. We call these essential genes, right? They're important. So what they're plotting here is they're plotting the percentage of essential genes in a group of genes that have a certain number of edges, right? So let's say they take the network and they say, give me all the nodes that contain 5 edges, and then within this group, what percentage of these contain essential genes? Okay, so all the nodes that have 5 edges contained about 22% of these genes were essential. But what you can see is that for genes that have a higher number of edges, the percentage of essential genes within that group increases. So what this means is that that's a positive correlation between a gene's likelihood to be essential and the number of edges that it has, right? So again, reinforcing our idea and quantifying it that the genes that have more connections, the proteins that have more connections, tend to be more essential. Any questions about this? All right, so that was yeast. For a lot of omics techniques, we start with yeast because it has a small genome, it has a small number of components, so it gives us a little chance to practice, right? And then what we usually do is take this to humans, right? So this is the human protein interaction, the first iteration of the human protein interaction network. These researchers actually repeated this many times, including more and more proteins that are to be tested. So in humans, they have tested 8,100 proteins interaction with itself in pairwise manner. So that is basically 65,000 interactions that are tested. They found about 2,800 interactions, and they plotted the network. So the network is really large. It looks like a hairball, so I'm not going to show to you. But I'm going to zoom in and show you one part of the network in which you can see a very highly connected hub here, which is called the EWS gene, which is a hub in this network. It makes connections with many, many other genes. And it's also a protein whose mutation is known to be causal to Ewing sarcoma. Okay, so again, you have a gene which has lots of protein-protein connections, and also has an important function in that is essential. All right, so these are protein-protein interactions, and you can find out which are the important genes and hubs, and you can look at this interaction as a whole, as a network. Is that any questions about this? All right. So there are other type of interactions that we can also measure. Here what we're looking at is we're looking at 2 genes and asking how related are these 2 genes' functions, okay? So for this, we use a method called synthetic lethality. So these are genetic interaction networks. So the previous one was protein interaction network. This is a genetic interaction network. So what we're doing here is we're looking for synthetic lethality. So this is defined as a combination of mutations in 2 or more genes leading to cell death, right? So whereas a mutation in only one of the genes does not. So this is also the basis of epistasis, which means that the effect of one gene is dependent on another gene. Okay, so effect of one mutation being dependent on another mutation. And this idea is related to genetic robustness. What that means is that in our genome, for a lot of essential functions, you have redundant genes. What that means is 2 genes actually kind of doing similar functions. So if you have a knockout of one gene, the other one still makes up for the loss of that other gene. Okay, so that's give us robustness. So imagine this is an airplane, right? And there are 4 genes, and let's say all of these 4 genes are the engines, right? And they're all doing a similar job, right? They're just running the plane. You have a mutation in one of these, and the plane shakes a little bit. You have another mutation of another one. You start screaming, right? And then this one knocks out, the pilot is screaming. And then this one knocks out, I'm not going to go there, right? So it's basically, this is kind of what synthetic lethality kind of assays do. So they knock out 2 genes at once and then see if you see a phenotype, okay? So, yes?
This is different than alleles where you have 2 copies of the same gene. These are 2 different genes. the organism will survive.
Well, it is similar, right? Let's say that you have two copies of the same gene. It is the same sequence doing the same thing. If you had a knockout of one of them, you wouldn't see any phenotype, right? If you have a knockout of another, and if this gene, when by itself is actually important, that will give you a phenotype. Let's say you have a pilot, right, and a co-pilot. They're doing exactly the same function. They're just taking the plane. You take out the pilot, the co-pilot takes the plane, and you take out the co-pilot, again, you're in trouble, right? So similar idea. All right, so how do we actually do this, right? This in yeast has been done many, many times, and so genetic interaction networks, the technique that is called is the synthetic genetic array analysis, so SGA analysis in yeast. So what in yeast the researchers have is they have a library of many, many yeast strains in which a single gene is knocked out. Okay, so in yeast, whenever you knock out a gene, you give the gene name, right, BNI1, and then you have the knockout, which is a deletion, so shown with a delta, right? All right, and then you have a bunch of genes that contain different knockouts. So let's say that you want to find out the genetic interactions between BNI1, and all of the rest of the yeast genes. What you do is you take a cell that contains this BNI1 deletion, and you mate it with another cell. These are haploid that contains the deletion of your interest. So you're going to test each gene one at a time in the knockout library. When you mate these and then you sporulate them and generate haploid phenotypes, haploid genomes again, 1/4 of your resulting yeast cells is going to contain both knockout, okay? So you're going to do some clever yeast genetics, which I'm not going to go through here, and then you're going to select for all of the strains that contain your double knockout. So knockout for this gene, and then knockout of each and every gene in the knockout library, okay? And then you're going to see, compared to a single knockout of this gene and a single knockout of your test gene, what is the phenotype of the double knockout, right? And the phenotype that we look usually is growth. So if you see the yeast cells forming a large, nice colony, you know that they're healthy, they're growing a lot, right? And if they produce a puny little tiny colony, you know that they're growing very slow, right? And if they don't form any colony whatsoever, you know that they're dead, right? So they are synthetically lethal. So the analysis is this. You look at this strain, you see a nice fat colony. So deletion of this by itself caused nothing. You look at this strain, which forms a nice fat colony. Deletion by itself causes nothing. And then you look at this strain, and if these two genes are synthetically lethal with each other, this double knockout is not going to produce you any colony, okay? So that's what you're measuring. And these are done by little robots that mix up these little strains together and prints them onto these large plates. So you can look at these synthetic interactions. For example, here are some strains that didn't grow on this plate, right? So here's a slow-growing strain. I kind of see a tiny little colony, but not so much. So then the scanners are going to take these images, and then it's going to convert the data, and it's going to tell you which two genes in the yeast genome are interacting with. And in that case, what you're going to do is you're going to take that data and, again, plot it as a network, right? So you're going to do a network analysis. So this is the yeast genetic interaction network here, shown as a nice hairball, right? And what we're doing here is that we're plotting the network analysis. So what are the nodes in this network? What are the nodes in a genetic interaction network? Genes. What are the edges? What are the edges? When do you put a line in between two genes? When they are synthetically lethal, when they interact with each other. Okay? So those are your edges. So what we're looking at is a zoom-in view of a few genetic interaction networks of interactions. And what you can see is, for example, they're color coding all the genes that are involved in amino acid biosynthesis and uptake in green. Okay? So what you can see is that the genes that are involved in the similar pathway seem to interact with each other, right? So they are closer to each other in the network, and they're away from other genes that have a different function. For example, genes that are involved in autophagy are basically interacting with each other more, right? So they're basically segregating by their functions. So if you do a similar type of this kind of analysis, and you have a bunch of genes that are involved in different pathways, you can look at these genes and then find out what kind of pathways are forming in these genetic interaction analysis. So how can this kind of network analysis help us assign gene function? So you did do this in yeast. You did a genetic interaction network. You formed the network. You looked at the little clusters of genes that have similar functions. And we talked about how even though we have genome sequences, we actually don't know what each gene does. In fact, more than half of the genes, we actually have no idea what they do, right? But these kind of network analysis allow us to make predictions. So tell me how you can use this network analysis to help assign or predict a gene function.
Which genes it's related to by synthetic lethality, then you can kind of take a guess at which pathways.
Exactly. Very good. So, for example, you have an unnamed gene, right? So you have a gene of unknown function that clusters right in between, clusters with all of these genes here. Then you can say that perhaps the function of this gene is related to this pathway, right? And it will give you a hypothesis to test, and then you can test whether that gene is involved in that function or not. Make sense? Any questions? Yes?
So in this pathway, you don't have to know the functions of the genes, you just have to know the genes and what they interact with?
Exactly. You don't need to know the function of the genes at all. But knowing the functions of the genes will help you assign functions to different clusters, right? So you know, okay, I know that from previous experiments that the genes that are in these clusters all seem to be involved in this function, and then it will allow you to guess this gene, who you don't know its function, may be related to the other genes, yeah?
Aren't we missing a whole lot of interactions of like two genes that interact, but when they're both knocked out, it's still not lethal?
Exactly. So this is like any biological experiment. When the data outcome is negative, you can't interpret it, right? That doesn't mean that whenever you do test two genes, they don't interact with genetic synthetic lethality. That doesn't mean that these genes are not interacting in some other way, right? Does that make sense, guys? That's why you need to do these network analysis using many, many different interactions. You look at protein-protein, synthetic lethality. You can do ChIP-seq and gene expression analysis, etc. Right. Any other questions? All right. Okay, so you can do this synthetic lethality and genetic interaction analysis in humans as well. But of course, we don't have a library of humans that have knockouts of each and every gene, right? So we do these in vitro, in dishes, using human cells. And so the idea is very much the same. You have a gene of interest, and then you look at a library of genes and see if they are interacting or not. So these are pairwise interactions, and you can choose a number of genes and then look at the interaction against another gene. Okay, so what you're essentially doing is that you're knocking down one gene, right? And then you're knocking down the gene 2, and then you're knocking down both genes, and then see what the phenotype is, right? The measurements. And so if you don't see no phenotype in one gene knockdown and another one, you see no phenotype, but with both of them, you see a phenotype, then you put these genes 1 and 2 as the nodes, and you connect them with an edge, okay? So how do you do this? The way that people have been doing this is they use RNAi. You learn this, right? You use small oligos, small RNA, to knock down the expression of a gene. So you can do RNAi against gene 1, you can do RNAi against gene 2, and then you can mix these two RNAis, and then you simultaneously knock down gene 1 and 2, and then look at the phenotype. So you can actually then measure a phenotype by looking at these cells, whether they're growing or not, etc. Okay, so that's the RNAi knockdown. So what other technology other than RNAi can you use this kind of in these genetic interaction network analysis in human cells? So RNAi is one way to knock down the expression of a gene. We've learned other ways in which we can knock down the expression of a gene. I'm going to give you a moment to recollect and recall some of the information. How can you knock down the expression of a gene in human cells? What kind of technique can you use to do this? CRISPR-Cas9. Be more specific. In the CRISPR lecture, we went through some methods to knock down genes expression.
You use the dead CRISPR-Cas9?
Very good. You use the method, which is we talked about, you fuse a repressor domain to the dead Cas9 protein, and then you can target that, the promoter of your target gene, right, and knock down the expression of this gene. If you wanted to adapt this to a genetic interaction experiment, you would have two different targets, right? You would simultaneously put the dead Cas9 with the repressor to two different target genes and knock down both of them and see if they interact by measuring the phenotype.
What kind of human cells do you use for this kind of array?
You usually choose whatever is the relevant to your analysis. For example, if you're...
Can they all grow in that kind of medium?
Yeah, so you have to adjust the requirements of your given cell type, right? And then do your experiment like that. For example, if you're interested in cancer cell proliferation in melanoma, right? So you could use some melanoma cancer cell line, right? If you're interested in just embryonic stem cells, you could use some embryonic stem cells. The human ones are going to be hard to come by. Yeah. Other questions? All right. Sorry, I'm kind of like, my voice is kind of bad, so I see some of you guys sleeping. I don't blame you, okay? It's all right. Don't worry about it. You listen to it again, and now you'll catch up, okay? All right. So, what are some other interactions that we can measure? We can measure expression correlation, okay? What do I mean that? All right, so for example, to understand the differences between a disease or a normal, or two different cell types, or even two different species, we can actually analyze their transcriptomes. And we can push it one step further, and we can look at how the different genes within a species actually interacts with each other, which is going to tell us something about how these two systems are different from each other, right? So we can compare the gene expression network in two species to try and figure out how these two systems differ from each other, okay? So for example, in this kind of a not so great experiment, but kind of ambitious, the researchers were trying to compare the human-chimp gene expression correlation using microarrays. And so they created a gene expression network. So this is the network in humans. And you can see that in this network, there's these 4 genes that make lots of connections. So these are the 4 hubs in this network. And when you look at the chimp, you see that one of these genes is still a hub, but another one is actually no longer a hub. And this one is no longer a hub, right? So the gene expression network between these 2 species are actually different. All right, so here's the question. In this network, the nodes are the genes, but what are the edges? What is connecting 2 genes to each other? So these are gene expression networks. The hint is, think how you measure co-expression. How do these 2 genes that are connected with the edge relate to each other? How do you draw those edges? What do those edges mean?
Synthetic lethality?
That's a genetic network. This is a co-expression network.
Like, if it's transcribed, would be like a little microarray analysis?
If their expression is correlated. What that means is that 2 genes, so what the measurements are here are, so you have a microarray analysis, and you have your gene A and gene B and C and D and E, and you do these analyses many, many times in many, many different conditions, right? So condition 1, let's say in nerve cells, when they're upon a heat shock, then it's muscle cells, and then you do plus serum, etc., etc. So you have all of these conditions, okay? So, and let's say this gene A goes up in this, goes up in this, go down, up and down, and this gene goes up, up, down, and this one goes up, down, up, down, up, down, nothing, down, up, etc., right? So what you can do is you can take these and then ask, what is the correlation between 2 genes expression, right? So these 2 genes are going to be well correlated because they are going up and down in the same manner across different conditions, right? And then these 2 genes are not correlated, A and C, very well, because once A goes up, C goes down, etc., etc., right? So what you can do is you can have a measurement of correlation amount of expression between these 2 genes. Why do we care, right? What is that telling you? What is that telling you is it's telling you that these 2 genes are required similarly. Remember how we talked about in muscle cells, MyoD co-regulates the expression of a bunch of genes that are required for muscle function? We know that the genes that are co-regulated, that means they are expressed together, tend to have similar functions, right? So you can say, okay, then I'm going to make the assumption that whenever I see 2 genes that are expressed and regulated similarly, they might have a similar function, and the amount of correlation that I see in between a whole bunch of conditions is going to tell me something about the strength of that correlation, that strength of that similar function, okay? So what you do is in gene expression analysis then, that you have your nodes here, which are going to be the genes, and the lines here that are connecting to the other gene is the correlation coefficient, right? Right? And if it's high, you're going to draw a line. If it's low, you're not going to draw a line. Does that make sense? Any questions? Yes?
How are the conditions chosen? Is it just like infinite conditions, or do they have a set number that they usually use?
So conditions, in most of these type of analysis, the conditions are chosen by the feasibility of the experiment, right? So the examples I gave, like heat shock and serum, etc., these are things that you can do, right? So whatever you can do determines the condition, and the number of conditions are usually determined by how much money you have, right? Because you have to pay to do these experiments. Make sense? All right. Okay, so some of the other type of interactions that you use to build networks are include such as microbe-microbe correlations. So you can look at the presence of one microbe and the presence of another microbe. If they co-inhabit in your sample, then their presence will be correlated, right? So you can make microbe-microbe interaction network. You can do microbe-metabolome interaction network, right? So the correlation between the presence of a microbe and a metabolome will connect these two nodes, right? And this will help you understand host-microbe interactions. And transcription factor transcriptome interactions will help you understand transcription factor mechanism, etc., right? So these type of network analysis in small scale, what that means is you look at few interactions with few genes, or large scales allow you to do to generate hypotheses about very interesting systems questions, right? So here's one highlight how this can be used. So in this example, the researchers are interested in doing a precision microbiome experiment to find out what are the microbes that actually maybe are related to resistance to Clostridium difficile. So this Clostridium difficile is a major cause of antibiotic-induced diarrhea, greatly increases morbidity and mortality of hospitalized patients. So these patients get these infections and they can't get treated. So they correlate with the loss of specific bacterial taxa with development of infection. So what that means is that whenever they see this infection, they also see a loss of a certain group of bacteria from that patient. Okay, so there is a correlation between the loss of this group of bacteria and this infection. So, so by treating mice with different antibiotics that result in distinct microbiota changes, then they can see that leads to varied susceptibility to Clostridium difficile. What that means is a specific group of bacteria loss is correlating with the infection. Okay, so if they can actually then find what these bacteria are, right? So they can perhaps provide these bacteria and fight against this Clostridium infection, right? So basically, they determine that this bacterium is associated with resistance to the difficile infection, and if you administer this bacteria to the mice, that enhances the resistance to infection, right? So you can actually treat the infection by putting another bacteria to the patient, right? And so that's one of the ways in which you can use these correlations to build a hypothesis and then test the hypothesis and see if this actually is causal, right? Any questions about this? All right. Okay, so that concludes today's lecture. Tomorrow we're going to have a 10-minute overview, and then after that we're going to have a review session. So make sure that you bring your questions, etc., to this session tomorrow, and I'll see you tomorrow, guys.